Let us do Brahmanan. Hello students. In the previous session, we discussed the genetic basis of cancer. All cancers are having some genetic basis, genetic region. There are some the cellular growth regulating genes. Mainly this can be categorized into two types, that is oncogene and tumor suppressor gene. The oncogene or tumor suppressor gene present in all the cells. In every cell these genes are present. In normal condition the oncogene is called cellular proto-oncogene. The proto-oncogenes they have the control over the cellular growth. They follow one principle, the contact inhibition. While cell division, when if that mass of tissue comes in contact with another tissue, it ceases the cell division. Even it follows the cell division or the growth of the tissue up to a certain period. It has the control, it controls the cellular growth. The tumor suppressor gene that suppresses the or prevents the formation of development of tumors. If any damage is there, it fixes it by repairing it, or it kills those tissue, those cells. We discussed the diagnosis, the symptom and treatment. The cancer can be diagnosed by the biopsy, tissue culture. By taking a sample tissue, it can be cultured and studied. The fine needle aspiration, it can be diagnosed by the CT scan, the MRI scan, the X-rays. Even monoclonal antibodies along with the radioisotopes are used for the diagnosis of the prostate cancer and the thyroid cancer. Cancer patients generally they become low weight, means they lose the weight, they get the disorder in digestion, and the open body part, the openings of the body, the bleeding is occur. Bleeding is shown from the openings of the body, like the nose or ear, mouth, etc. Cancer can be treated by 4 ways. In 4 ways, cancer can be treated. First, complete surgical removal of the cancerous tissue or the tumor. The radiation therapy, where the cobalt 60, the radon 220 or iodine 131 are used, or the chemotherapy, anti-cancer drugs are given, or the immunotherapy. In immunotherapy, mainly we can say the interference. Even in chemotherapy also, the interference are given, are introduced to the cancer patient. As the interference are the immune product, we can say in immunotherapy interference are used, or that monoclonal antibodies along with the single or along with the radioisotopes are used for the treatment. In this session, we are going to discuss acquired immune deficiency syndrome, AIDS. In the previous session of immunity, we discussed the disorder of immune system is characterized by overactivity or low activity of the immune system. The overactivity results into hyperimmune response, whereas the low activity results into the impaired immune system due to the deficiency. Deficiency that can be immune deficiency disorders, can be due to the malnutrition, due to lack of the nutrition, or because of the infection. The acquired immune deficiency disorders that leads to decrease in the ability of the body to fight against an infection. Okay, this immune deficiency disorder are of two types. They are primary and secondary. The primary immune deficiency disorder are due to the genetic cause. Whereas the secondary immune deficiency disorder are due to the acquired cause. And this is caused due to the malnutrition or infection, or infection. Example of autoimmune disorder that is due to infection is AIDS, acquired immune deficiency syndrome. Syndrome means also disorder. If acquired immune deficiency disorder is there, then it results into the reduction in helper T cells. And this leads to loss of natural defense against viral infections. Am I clear? Then if the AIDS occurs due to the infection, the body loses the natural defense against the viral infection. When the immune deficiency disorder occurs, then the deficiency of, as I told, malnutrition or infection we are saying, or genetic, it can be genetic or acquired. Acquired immunity, as we know, it is AMI or CMI, but here AIDS is a kind of, it is a disorder of cell-mediated immunity, CMI. Okay, and acquired immunity played by B lymphocytes and T lymphocytes. And T lymphocytes, they recognize after on the entry of pathogens in CMI. I am showing you over. In CMI, the T cells differentiate into cytotoxic T cell, helper T cell, CD8 plus, CD4 plus, the memory T cells, suppressor T cells, and these T cells, they fight against the foreign substance, the CD4 or helper T cells. These cells usually either they stimulate the CD8 to kill the pathogen or antigenic substance, or it induces the B cells, and B cells produce the antibodies. So, in the acquired immune deficiency syndrome, it leads to this way. Helper T cell are destroyed, then they are not able because of the deficiency of helper T cells, the B lymphocytes, B cells are not stimulated for the production of antibodies. Let us go through the history. First, AIDS-1. AIDS was noticed in USA among homosexuals, those who are having the sex in between the same gender. In 1981, first AIDS was noticed in India. In India, where? In India, in Chennai, in the prostitutes of Chennai. In 1986. Now come to the discovery. This is also, so how it was discovered and who discovered? The Francois bear Sinosi. Francois bear Sinosi and Luc Montagnier. G remains silent. You can say to remember the spelling Montagnier, you can. Okay. This is bear Sinosi. The Montagnier received the Nobel Prize for medicines or physiology for for physiology and or medicines for 2008 for AIDS research and prevention. Okay, Francois, Bert Sinosi and Luc Montagnier, they received the Nobel Prize for Physiology and Medicine. In that year 2008 for Physiology or Medicines, even who received also that Harold Jourhassen received that Nobel Prize for Physiology and Medicines. They discovered, actually Luc Montagnier along with the Francois Sinosi, Bert Sinosi received they discovered the virus called HIV that causes AIDS. The pathogen. The pathogen that causes the AIDS, that was previously known as human cell leukemia virus 3. What I told? Human cell leukemia virus 3. Who named this? By Americans. It is named so by Americans. Then the SCLV3 is renamed to its official name HIV, that is by the International Committee on Viral Nomenclature, ICVN. What is the full form of ICVN? International Committee on Viral Nomenclature. ICVN renamed that SCLV to HIV. HIV is the official name of AIDS virus or virus causing the AIDS. Okay. What is the full form of HIV? Human immunodeficiency. Human immunodeficiency syndrome. This is a retrovirus. Retrovirus, this is a retrovirus belong to the retrovirus family Retroviridae, belongs to family this HIV belongs to the family, here I am writing family Retroviridae, and the genus lentivirus. The genus is lentivirus. Okay, what I told, the HIV is the pathogen. HIV in the year HIV was the official name of the virus causing AIDS by ICVN, International Committee on Viral Nomenclature, in 1986. It is believed that the AIDS virus, that is HIV, may passed from monkeys to monkey host to human of Africa in 1960s. Okay, so now let us discuss the structure of HIV. The structure of human immunodeficiency virus that causes the AIDS. Structure of HIV, human immunodeficiency virus. As we discussed, the HIV belongs to the retrovirus family Retroviridae, and genus also it is told subfamily. Genus is lentivirus. The lentiviruses, they are having the specific characteristic is protein capsid. How it is found? Let us say the lentiviruses, the HIV, human immunodeficiency virus, having the 2 single-stranded RNA. The genetic material is 2 single-stranded RNA. That is surrounded by the protein capsid. That is viral protein capsid. Capsid is covering. Capsid is nothing but covering. So, it is the protein covering made up of P24. It is P24, within which the viral genomic material is there? 2 single-stranded RNA, and again it is covered by another protein layer, that is the spherical protein matrix, and again this is covered by, surrounded by the lipid envelope. The lipid envelope is of host origin. This lipid envelope is of host origin. It is produced when the viruses are dropped out from the host cell. This envelope is developed when the viruses, they are produced, dropped out from the host cell, and this lipid envelope again is consisting of the spikes, okay, glycoprotein spikes. Showing you something, the glycoprotein spikes are again of consisting of two glycoprotein. Okay, this is hook-like extended from the lipid layer, and this is the spike. This green part, green glycoprotein spike, not it is actually green colored, I have shown over here. Okay, these are the spikes, glycoprotein spikes. What are these? These are the glycoprotein spikes, which helps to anchor the host cell. Okay, host cell. You see, this is the glycoprotein is represented as GP. So this is made up of GP120, and this is the blue one. Suppose this is, the stalk is like this, the blue one. Okay, I am not showing or making at least. Okay, the blue one is made up of GP41. Okay, now see, this is a protein capsids, and this is the lipid envelope. What is this? This is lipid envelope. This one is the internal protein capsid. Internal capsid, internal protein covering, you can say protein covering or capsid capsule like covering structure. Capsid, this is P24. This is P24, and the external protein covering is made up of P18. This is P18. External protein covering P18, also known as matrix protein. Matrix or the matrix protein. Outer, here it is better, inner and outer. Outer protein capsid. Okay. Now the enzymes present over here, they are the reverse transcriptase. The RNA, these are the single stranded RNA. What is this? This is genomic RNA, single stranded genomic RNA. And there are two number, these are present within the inner protein capsid or inner protein covering, that is RNA, single stranded RNA, along with the reverse transcriptase and integrase. These two are reverse transcriptase. It helps in reverse transcription in the host body. Reverse transcriptase. AC is added, means you can understand it is an enzyme. And other one is the invertase. These two, I am labeling this side, or okay, let us do this side only. These are invertase, sorry, integrase. It cleaves the genetic material, and even some other proteins are there and enzymes. Here are the some proteases. Okay, some more proteases. Protease in between the capsid and matrix. It is simply inner protein covering is called capsid. Outer protein covering or outer protein capsid simply called matrix. Okay, it is called matrix. So in between the capsid and matrix, there are some proteases and some more enzymes are present over here. Remember, these are some more enzymes. These protein coverings are viral origin, but the lipid envelope, this lipid envelope is of host origin. These are produced when the viruses, these viruses are dropped out, because as we know the viruses, they divide inside the host cell. When they are dropped out, when the viruses are dropped out from the host cell, they are enveloped, the protein capsulated viruses, they are enveloped by the lipid layer. Okay, and these lipid layer are having the glycoprotein spikes. Again, these are made up of two parts, that is glycoprotein, that is GP120 for anchoring, to anchor or anchoring to host cell, and this is the stem part GP41, that is holds the GP120 along with the lipid layer, that is the envelope of the virus. Okay, and this is the structure of HIV. Let us discuss how these viruses, they are transmitted and they infect the host cell. When these HIVs, when the HIV infect a portion, infect a healthy person, then HIVs attack the T cells. But however, these HIVs, the human immunodeficiency viruses, they infect the macrophages, which are the tissue monocytes present in the tissue part. Okay, draw this level diagram. Remember one thing, that here the RNAs are with some, RNA is drawn like this, viral genome. RNA are the viral genome. Where I have written, not labeled. Okay, yes, reverse transcriptase RNA. Let me draw here. The viral genome. They are attached with some nucleocapsid. They are still, the RNAs are covered by the nucleocapsid. Here written single stranded genomic RNA. I have written here. Okay, they are covered by the nucleocapsid. Nucleocapsid, these are the nucleocapsid. This diagram is required for the conceptual clearance, to clear your concept. But when it is required to be drawn, when you get this question AIDS, that is structure of AIDS, in the 3.5 question of the long question, 3.5 short note of the long question. So remember, there are the three places the capsids, protein capsids are there, capsid covering, capsule like. So here the RNAs are also covered by nucleocapsid, means directly the nucleic material is covered by protein capsid. Okay, and this one is the called inner protein capsid or covering. protein capsid or covering. This is called matrix protein. This one is matrix protein or the outer. This one is the outer protein covering or matrix or outer protein capsid. Matrix or simply matrix protein also called. Okay. This is HIV, structure of human immunodeficiency virus. Structure of human immunodeficiency virus. How that these viruses infect the host cell, let us see. Write down the points of structure of HIV. For structure of HIV, okay, belongs to family Retroviridae, the genus lentivirus. It is roughly spherical, measuring about 60 nanometer in diameter. Two single-stranded RNA. Okay, transcriptase and integrase. Also, the nucleocapsid are there. Conical inner capsids are P24. The outer matrix proteins are a P18. The glycoprotein spikes. Are of GP120 and GP41. Okay, and proteases and some enzymes present in between the capsid and the matrix protein. Now, let us discuss the mode of action of AIDS or HIV virus. The mode of infection. How the HIV infect the persons, human beings or other animals also. First, they enter into the bloodstream. That is, enter into our body either through blood or through the semen, through the tissue fluid. That is specifically through blood or semen. And then they attack the macrophages and T lymphocytes or T cells. Then they anchor to the macrophage and they empties their content, genomic content, into the host cell, that is macrophage or the T cell, and within that the RNA, the genomic content is the RNA, single-stranded RNA. Only the RNAs are emptied into the host cell. The protein capsids are left outside the cell, and these RNAs, they undergo reverse transcription in the presence of reverse transcriptase enzyme, which is present along with them, undergoes the reverse transcription and become viral DNA in the presence of reverse transcriptase using all host cell machineries. How? They attack the macrophages and helper T cells, and their viral RNA, they undergo reverse transcription to become viral DNA using all machineries of host cell. Uses all machineries of the host cell. Then transformed to that DNA. Okay, that viral DNA means that viral RNA becomes the viral DNA. Then that viral DNA incorporates with the host DNA. Let me draw the diagram to make you understand. Suppose this is the macrophage. This is a macrophage. Okay, then here are the free virus HIV with the RNA. Okay, now when it comes in contact, their glycoprotein spike attached to the receptor present on the comes and attached with the receptor present on the cell membrane. The host cell, this is the macrophage. Cell membranes are having the specific receptor cell. They come and contact with the host cell receptor. Host cell receptor. Then what happens? You see, I'm showing the next step. RNA enter into the cell, and within the host cell the nucleus is there. This is the host cell nucleus, and within which, suppose this is the host DNA, you think. Suppose this is the host cell DNA, and this RNA undergoes, this is RNA, viral RNA, vRNA, I am writing vRNA. vRNA undergoes reverse transcription and becomes vDNA. That is, from single stranded it becomes double stranded. From single stranded it becomes double stranded. This is the viral DNA. Then viral DNA incorporates with the host DNA. So what happens? See, now this is incorporated, the viral DNA is incorporated with the host DNA. This is host DNA and this is viral, that DNA is incorporated. So this is transformed DNA. So this the viral when it is incorporated in the host DNA, it can produce large number of RNA. So it produces the large number of RNAs, viral RNAs are produced, and these viral RNA synthesize the viral protein. Synthesize the viral protein. Suppose these proteins are like this. Okay, viral proteins are capsids. Capsids are developed, and these capsids they surround the RNA. Okay, when they bud out, they bud off, means pinch out from the host cell, then they are enveloped by the bilayer lipid. Actually, then when they bud out, as if this is the bilipid layer, they punch out this is how large number of viruses are produced within the body from the host cell, and this is why the macrophage, that you see macrophage act like a virus factory. And this is why it is called macrophage becomes the virus factory. Macrophages, they produce viruses and act like the virus factory. What I told, that viral RNA undergoes the reverse transcription producing the viral DNA. Viral DNA incorporates with the host DNA, and transformed DNA produces many viral RNA, and viral RNA synthesize the viral protein, then viral protein along with the RNA. RNA is surrounded by the viral protein and gives rise the viruses, and they bud up with the lipid envelope. And this is how the macrophages produces a large number of viruses, becoming the virus factory. Simultaneously, the viruses, they attack the helper T cells. Okay, there also they produce the more viruses, and thus the T cells decreases. Helper T cells, okay, helper T cells attack the helper T cells, and the helper T cells decreases, and thus the infected person suffer from during this period. During this period, the infected person suffer from fever, diarrhea, and weight loss. And by this time T lymphocytes are decreased, so the body is easily infected by other viral or bacterial or fungal toxoplasmic infections. Okay, write down all this.